My name is Dr. John Elkins, and I am the academic head of the Centre for Medicines Discoveries, Biotechnology and Lead Discovery Facility at the University of Oxford. New potential drug targets are continually being identified by scientists here in Oxford and around the world, and most of those drug targets are proteins. We support early-stage drug discovery by finding molecules that modulate those target proteins. These molecules can be traditional small molecule drugs, but also linear or cyclic peptides or miniaturized antibodies. We want to have molecules that are potent and specific to the target, so that when they are tested in cellular or animal disease models, we can get the earliest possible indication of whether modulating the drug target will have the desired effect on the disease. We can then support the further development of the molecules.
My role is to develop early-stage drug discovery projects, which often requires the development of novel protein reagents. All proteins are unique, and producing them requires a bespoke approach, balancing the characteristics of the protein and the end goals of the project. We can design proteins with a variety of tags or alterations as required to produce the high-quality protein reagents needed to enable drug discovery projects. From our 16-year history as part of a world-leading structural genomics centre, we have the experience of having produced thousands of different proteins, and this allows us to design and develop expression systems for almost any protein drug target. In our facility, we can produce proteins in bacterial cells, insect cells, or mammalian cells. For developing new protein expression systems, we utilise high-throughput cloning followed by parallel, miniaturised test expressions to find those constructs that produce the highest quality protein samples, and from there we develop optimised purification strategies.
I'm a scientist specialising in assay development and small molecule screening. We design assays to test the effect of inhibitors on target proteins. These assays allow us to screen for inhibitors and characterise the hits we find, including measuring binding affinity or binding kinetics. Sometimes we want to inhibit a protein's enzymatic activity, but in other cases we may want to disrupt or even stabilise a protein-protein complex. We use a variety of instruments to conduct high-throughput screening, which can range from fluorescent assays or label-free assays to mass spectrometry. Using our miniaturized assays and robotics, we can screen libraries of tens of thousands of small molecules. Alternatively, we can screen fragments. These are low molecular weight compounds that have a higher probability of binding to the target protein. Although fragments may have a lower initial binding affinity, they can then be optimized into potent inhibitors. The robust and reproducible data we generate is crucial for advancing initial inhibitors into molecules suitable for preclinical evaluation. Beyond identifying inhibitors, we also study their functional effects on proteins. For instance, understanding how inhibitor binding affects protein stability or oligomerization can be key to optimizing inhibitors. And, apart from small molecule inhibitors, we can use similar assays to characterize other types of modulator, such as cyclic peptides or nanobodies.
All of our projects are collaborative, and we work closely with many scientists and clinician scientists to drive research forwards. We're always open to new collaborations and happy to discuss how we can help advance your project using any of our platforms. We would love to hear from you and help you advance your research from bench to clinic.